Meet Hendrik Poinar. He's a bit of a mystery man, as in he likes to solve them. And he's part time traveler, as in he likes to dig up the past. Think Doctor Who meets Indiana Jones. Hendrik Poinar is on an extraordinary quest. His mission? Solve one of the greatest mysteries of science, a mystery that has eluded researchers for more than 6 centuries. Unlock the secrets of the killer disease that caused the Black Death and wiped out more than 50 million people, half the population of Europe. It's a daunting challenge. To help him, Poinar has assembled an international team of experts. There's a lot at stake here. If they can solve this mystery, they'll gain knowledge that could help us fight deadly infectious diseases today and potentially save millions of lives. And if you thought the Black Death was a thing of the past, think again. It still seeks out victims to this day.
Sorry, there's no one here to help you.
Yeah.
Hendrik Poinar is an evolutionary biologist, which means he studies the nature of how we humans got here and where we're going. Hendrik's childhood dream was to travel the world and travel back in time. He's achieved both through his work, which includes flying to places like Russia in search of woolly mammoth fossils.
I am face to face with a mammoth. I mean, how many times can you say that? These boyhood dreams as a kid when you think about being up next to an extinct animal.
And of course, you're thinking of all the DNA that's...
Yeah, I see genome, I see chromosomes.
Poinar is also one of the world's leading experts on ancient DNA.
No one imagines that there's actually something still hidden within a bone that's been buried for a few thousand years or 100,000 years, let alone the possibility of resurrecting it or bringing it back to life. I mean, that's sort of completely bizarre. It's like a time machine. Yeah, it's a kid's dream.
The idea of using ancient bones as a time machine inspired Poinar to embark on a fantastic adventure. He knew from his previous work that ancient DNA is filled with scientific secrets. So he set out to try and solve one of the greatest mysteries of all time. What caused the Black Death, the 14th century plague that decimated Europe?
So over the course of this 5 years, you have this epidemic that begins relatively localized and gets bigger and bigger and bigger. And it doesn't seem to be particularly selective. It still seems to kill indiscriminately, at least in the early stages. You know, we have no comprehension of what the horror and panic must have been like.
Since ancient times, Italy has been at the crossroads of trade between Europe and Asia. In the 1340s, it was the first European country struck by the Black Death. For Black Death hunters, Italy is ground zero. That's why Hendrik is here. Several Italians wrote detailed chronicles about the devastation. Hendrik's come to track them down in a search for clues. Clues that he hopes will help him unlock the deadly secrets of the disease. Hendrik is heading for Pistoia, a small town between Florence and Pisa. At Ospedale del Ceppo, he meets with local historian Lorenzo Cipriani.
Lorenzo.
Hello, hello, I'm fine, and you?
Good.
Oh, great, welcome in Pistoia.
Thank you very much.
Oh, great.
Lorenzo takes Hendrik to a maze of tunnels that run beneath the hospital.
So can you tell me, Lorenzo, a bit about these tunnels here?
Yeah, there are some very, very ancient stones in the bottom part. You can find some stones from the 13th century, exactly 12.
This was once the site of an ancient river. There are stories that bandages and implements used to treat Black Death victims were dumped here, no doubt helping to spread the disease.
So how do you think Black Death came to Italy?
We know, according with the description of Villani and other chroniclers, that came from the east, from India, from China, and then spread very quickly in our country.
Giovanni Villani was one of the most important chroniclers of the Black Death.
They began by spitting blood and later would die. Some straight away, others after a couple of days. It happened that the ones that took care of the sick would get infected with the same symptoms and consequently died.
Death came within 2 to 7 days, a final escape from the constant vomiting, diarrhea, delirium, and blackened extremities.
After the Black Death swept through, how many people did it wipe out? What percentage?
50% in the countryside and 60% in town.
60%?
Yeah, exactly.
Imagine every other person in the population getting sick and being dead within a week. Right from the time it first appeared, scientists have tried to determine the cause of the Black Death. Some have even come close to solving the mystery. The prime suspect was bubonic plague, caused by a bacteria called Yersinia pestis, also known as Y. pestis.
Some people in France, Raoult and colleagues had shown that Yersinia pestis was present in some of medieval samples, and other people had tried to replicate those findings and couldn't get any DNA or any results to indicate that. Yeah, so you were left with this hole, basically saying, well, we have anecdotal evidence, no replication. What do we believe?
To prove beyond a doubt that Y. pestis is the killer, Poinar needs to find a sample. He knows that the only place where the bacteria might be present is in the bones of victims. But documented Black Death grave sites are rare. However, there is one place where Poinar believes he might find what he's searching for. From Italy, the Black Death spread throughout Europe. It moved by land and sea along the well-established trade routes. It crossed the English Channel and quickly closed in on England's capital. Knowing it was only a matter of time before the plague arrived, London's city fathers took action. They built a massive burial site just outside the city wall in East Smithfield.
There was the foresight that lots of people are dying. Parish churches are not going to be able to cope with the amount of people dying. And so they then actually prepare burial grounds that can then cope with this catastrophe.
In the 1980s, when the area was developed, the Black Death victims were discovered and brought to the Museum of London. Curator Jelena Beckfelak oversees more than 20,000 sets of human remains housed here.
This is one of the individuals from the East Smithfield Catastrophe Cemetery site, and it's a female, and we're able to assess sex by looking at the pelvis, but we'd also be looking at other features as well. So I'd be looking on the surface here to see, could I see anything like infection? Have there been any fractures? Looking at the joints, is there any joint change? Looking at this individual, she's a young individual, she's probably only in her early 20s, and I can't see any other types of pathology actually affecting her bones. Something that killed her very quickly on this time, because we have the archaeological context. We were saying it's from the Catastrophe Cemetery site, it's the Black Death. And that's what makes this site particularly unique, is it's the only site that I'm aware of that has actually been archaeologically excavated that you can categorically say is a Black Death.
Poinar believes that these skeletons might contain DNA from the bacteria that caused the epidemic. To help him search for ancient Y. pestis, Poinar recruits one of the best bone detectives in the world, Sharon DeWitt.
So how many people are in here?
Probably now getting on for about 20,000 individuals.
Wow.
So yeah, phenomenal collection. Over 2,000 years history of London in here. So marvellous. We've got one maybe here that you might like to look at.
Oh, yeah, that looks really promising.
Sharon is an expert in human osteology. That means she can identify every bone in the human body, even from a fragment. But in this case, Sharon's focus is on teeth, because the pulp inside them is the most likely place to find blood. If there's any DNA, this is where it will be.
We're interested in obtaining samples of teeth because any pathogens that would have been in the blood system would also be in the dental pulp after death. And in the tooth, that pulp chamber is surrounded by dentin, which is hard tissue, and then that is surrounded by enamel, which is very highly mineralized, as opposed to bone, which is a lot more porous than a tooth is. And what that means is the teeth are going to provide a better, more protected environment for any DNA that's inside the dental pulp.
Sharon's selections are immediately shipped to Poinar's headquarters, the ancient DNA laboratory at McMaster University in Canada. Grad student Kirsty Boss is recruited and given the most important assignment she's ever undertaken: see if she can find a fragment of the pathogen suspected of causing the Black Death, Y. pestis. Kirsty starts by accessing the inner pulp chamber of the tooth in search of dried blood. If there is any trace of the pathogen that killed this person, it will be in here.
We then carry the powder through a process called a DNA extraction, where we isolate the DNA from all other molecules that are present in that powder, like lipids or proteins, other minerals that are present. So at the end, we get a purified dollop of liquid, which is called our DNA extract. And that is the liquid that will contain the pathogen that killed that individual.
Kirsty's dollop of DNA extract is more like a swampy cesspool of DNA. Human DNA, plant DNA, DNA from the earth and the animals that were in it. What she wants to find is a fragment of Y. pestis DNA and isolate it from everything else. It's a formidable task. Long days of trial and error become weeks. Weeks blend into months. It's like trying to find the proverbial needle in a haystack.
It's actually more than a needle in a haystack. You can think of it more as one grain of sand in a giant football field is maybe more of an appropriate analogy.
In the end, it takes over a year, but Kirsty's perseverance finally pays off. She discovers trace amounts of Y. pestis and successfully isolates them. Definitive proof for the first time in history that this bacteria is responsible for the Black Death.
And that was a very exciting moment for us when we actually found the presence of the DNA in the teeth, because we didn't know at the outset that we would be able to do that. It was a very good moment for us.
Even though it is an historic discovery, for Poinar and his team, this is only the start. Knowing that the Black Death killer is Y. pestis isn't enough. To unlock its secrets, they have to reconstruct the entire Y. pestis genome, its complete genetic identity.
We want to know how the bug has evolved. We want to know what the changes were that possibly caused it to be so virulent. So I wasn't interested in being able to say, well, we also see that Yersinia pestis is in these skeletal remains. Yeah. No, I wanted to get that genome so we could be able to address something interesting about the past and the possibility of what that means towards the evolution of pathogens and virulence in general.
Inside every cell lies its DNA. Y. pestis DNA is composed of 4.6 million pairs of chemical letters. These letters make up its genome, the entire blueprint that reveals how it functions. After months of work, Kirsty Boss had discovered just a few small fragments. Those fragments were tiny, none more than 50 letters long. If Poinar and his team can find the rest of the missing pieces and reconstruct the entire genome, they will have a powerful tool. It will allow them to discover not only what made Y. pestis so virulent, but also how it can be fought today. There is just one problem. Something like this has never been attempted before. There's only one scientist in the world who can help, and he's in Europe. Kirsty takes her Black Death teeth and Y. pestis fragments and flies to Germany. She meets with paleogenetics expert Dr. Johannes Krause and recruits him to the team. Johannes draws on his experience working with Neanderthal DNA to see if together he and Kirsty can achieve the impossible.
So the biggest challenge was that only a tiny proportion of the DNA that you get from a medieval bone is actually from the pathogen. And since we wanted to try to reconstruct the entire genome of this bacterium, we had to find ways to pull out the fragments that belong to the genome from this complex mixture of DNA that you get from the bone. Seems to be like 0.5 milliliter, so just the right amount. Just about what we put in, you get out.
Good.
Perfect.
Excellent.
So, take it, let's get it. Sequence.
Johannes uses a revolutionary technique that hinges on the fact that all DNA is constructed of two strands that are attracted to each other. Johannes exploits this natural attraction by splitting the modern strain of Y. pestis into single strands. He then uses one of those strands as a magnet to fish out all of the matching fragments of ancient Y. pestis from Kirsty's DNA extract. The next step is to put the fragments into a DNA sequencer.
And how long is it into the cycles at this point?
Well, right now we're at cycle 132, so we still have another 70 to go. So it'll take several days.
The sequencer analyzes Kirsty's fragments and converts them to billions of pieces of raw data. The final piece of the puzzle is to reassemble the fragments in the correct order. They load all the information into another computer and watch and wait.
It is really amazing how nice this looks like. You guys did a fantastic job. Just a fantastic job.
Thank you.
In the end, the team does it. They reconstruct the whole genome of an ancient pathogen, Y. pestis, something that has never been achieved before.
Well, we demonstrated for the first time that it was possible to reconstruct an ancient pathogen from archaeological remains, and that was an incredible methodological achievement.
That was completely, jaw-dropping, awe-inspiring, that we had the possibility to not only go back into the past in this sort of artificial time machine that we'd created and study humans and their DNA and what that meant, but we could actually now study the pathogens buried within these humans that lived during that time. Phenomenal, unbelievable.
But solving one mystery simply leads to another. When the team reassembles the Y. pestis genome, they discover it has mutated. Out of 4.6 million base pairs, there are 97 differences between the ancient and modern versions of this deadly killer. Those differences could prove to be critical in understanding the disease. Both the Black Death and its modern-day counterpart, bubonic plague. Decipher their role, and Poinar and his team may be able to help prevent future outbreaks. But for this man, it's too late. What he thought was a case of nasty flu turned out to be something much more deadly. The archaeological site at the church of San Pietro a Pasiveri is generating tremendous excitement. After years of digging, a mass grave has been discovered. An hour west of Florence, this was once part of a monastery that dates back to the Middle Ages. That means there's a good chance there are Black Death victims here.
We found 7 individuals this season, I believe. 3 female, 2 male, and 2 juvenile burials.
Even though there haven't been any Black Death victims discovered yet, there are still several more layers to be excavated.
Dated to the 19th century.
Hendrik Poinar is here because he needs more Y. pestis DNA to solve the riddle of the 97 changes that occurred between ancient and modern Y. pestis.
Help preserve
It's very likely that there are genes that we just haven't identified, and we now need to find those, along with understanding how those single changes affected the virulence are sort of the outstanding questions for us. Because if we can sort of predict which of those changes will have dramatic effects on virulence or its ability to infect a new host, those are the ones we want to understand.
Make those breakthroughs, and Poinar could help stop a future outbreak of this disease. It's a critical quest because the bubonic plague still claims lives to this day. Paul Gaylord lives in Prineville, southeast of Portland, Oregon. Paul used to be a welder, not anymore. A year ago, Paul lost all his fingers and toes. Paul's troubles began when he tried to help his cat.
The cat had been missing for a few days, and he just showed up. It was at night and it was dark out, but he had a mouse stuck in his palate. And a friend of ours and I tried to pull it out, and that's when we got bit. And it was just a couple of days later, I already had a fever.
Paul ended up in intensive care.
And then in the intensive care, the doctor walked in and said, "I think you have the bubonic plague."
I knew the cat had died, so I knew I was in for something, but who would think the bubonic plague, you know?
If the modern plague is diagnosed within 2 days, it can be stopped. By now, Paul was beyond this point. His case was immediately referred to the Centers for Disease Control at Fort Collins in Colorado.
So when we first heard about Mr. Gaylord, he was extremely ill. He was basically on death's door, and his body was doing everything it could to survive, but he was extremely, extremely ill.
Paul soon lost consciousness, was isolated, and put on full life support.
It was touch and go for a long time. Many of his physicians were not sure whether he would make it.
After 27 days in a coma, Paul's wife Debbie was faced with an impossible choice.
The doctor told us that we had to make a decision to unplug him or not unplug him, so we did. We made the decision to unplug him, and he woke up.
Although Paul regained consciousness, he wasn't out of danger. He was still suffering the ravages of the disease. His fingers and toes had turned black, he had buboes, swelling of the extremities, and dry gangrene, classic symptoms of bubonic plague.
We didn't expect you to live, and you fooled us all.
You have?
It's the CDC's job to guard against an outbreak of bubonic plague. So it was critical for them to discover exactly how Paul contracted the disease. They started by analyzing DNA samples from him and his cat.
In this case, we'd like to know if Paul got the infection from a flea, in this case a cat, or some other source. So by comparing the genetic profiles, we've been able to identify similarity between strains from those two sources, and Paul's and the cat that bit him are identical patterns.
There's no doubt Paul's cat was the culprit. That's not surprising. Bubonic plague in humans is rare, but in animals, not so much. The high plains and dry heat of the southwestern United States make it the perfect breeding ground for one of the cutest killers known to man. In North America, this is bubonic plague headquarters. And this is public enemy number one, the prairie dog.
So what I want you to do then is just to scatter out here, just sample as many holes as you can, see if this colony has any evidence of plague in it.
To guard against an outbreak of bubonic plague, CDC biologists at Fort Collins continually monitor the prairie dog population. It's a job that has become even more important since Hendrik Poinar's discovery.
But the fact that we now know conclusively that the Black Death was caused by Yersinia pestis, to me, is really important. It says, yes, that we have to remain vigilant. We have to keep an eye on this bacterium in nature because it does have the potential to cause outbreaks, and it does still cause outbreaks in the developing world, and those outbreaks are accompanied with a large percentage of fatalities. It's still an incredibly dangerous disease.
What Ken Gage and his team are on the lookout for is a prairie dog die-off.
When they die, what happens is their fleas have to find a new host. And that's when it's really dangerous for people. Those fleas will jump off that dead animal. And unfortunately, it may not be another rodent, it may be a human. So for us, the warning sign of increased human risk is really the occurrence of these, what we call epizootics, which are epidemics among animal populations.
There are lots of prairie dogs here, which is a clear sign that there hasn't been a die-off. That means there's no immediate threat to humans. But it doesn't mean there's no threat at all. Bacteria like bubonic plague are constantly mutating. What Ken Gage's team is watching for is a change in the genome, a change that could signal trouble. And to search for that, they need to gather fleas.
Shaking it up here, boss, shaking it up here. What movie that's from?
I have no idea.
You never seen Cool Hand Luke?
I am a baby still.
Famous movie.
Once captured, fleas are individually numbered and brought back to the lab. First task is to identify whether the fleas have eaten recently. Adult fleas thrive on blood, and that's where the bubonic plague is carried.
We are specifically looking for plague, Yersinia pestis, in the samples that we are collecting. I wanted to see if they have nothing in their guts or red in their guts. That might give me an indication that there might have been a host in their proximity recently. If you have the wrong flea on the wrong host, that could indicate a die-off in the area, which would then make us a little more concerned, especially based on public proximity. So it's important to know what fleas we have, what they came off of, and where they came from.
Once they're logged into the database, the fleas are sent to diagnostics.
What Shanna is doing is processing the fleas that we collected in the field. We've added a little bit of broth, and she's using a Teflon pestle to disrupt the flea exoskeleton to release the gut contents.
Disrupting the flea exoskeleton is science speak for crushing the critter.
Once that's completed, then we can take a portion of that liquid and plate it to see if we can detect plague.
The CDC is constantly checking the genome of Yersinia pestis to ensure it hasn't changed. Now, thanks to Hendrik Poinar, they have a better understanding of how pathogens evolve. By reconstructing the ancient Y. pestis genome and comparing it to the modern one, Poinar discovered 97 differences. If one of those was responsible for making the disease so virulent, Poinar will give CDC biologists a specific target to keep an eye on. In effect, an early warning system.
Well, the work that Hendrik's doing is very interesting because it enables us to see how organisms change over longer periods of time and get a better idea of what we might expect in the future. And the challenge is to try to figure out how those changes are linked with virulence, so what the risk to humans and animals is.
Every year, there are 2,000 cases of bubonic plague worldwide. Early detection is critical to avert the kind of tragedy faced by Paul Gaylord. To enable a quick diagnosis, the CDC has developed a rapid assay, similar to a pregnancy test.
Well, one of the challenges with plague is rapid diagnosis. You have about a 48-hour window from the time of signs and symptoms to treat someone. After that, the likelihood of survival goes down quickly. So what we've developed is an assay that we can take a sample from a patient and within 15 minutes have a diagnosis of plague or not.
Paul Gaylord survived the bubonic plague. But if a deadlier version of the bacteria emerged, the outcome for tens of thousands of people could be very different. Paul may never work as a welder again, but the self-proclaimed luckiest unlucky man in the world is philosophical about it.
Don't give up. You got to know life's worth more than your limbs. You can still have a good life afterwards, and you know, I've always liked a challenge. This isn't exactly what I had in mind, but I'll take it.
The bubonic plague that struck Paul is not easily passed from person to person. Not so for ancient Y. pestis. Poinar and his team believe one of the 97 changes could be the reason the ancient disease was so virulent. But they also have another clue. They think ancient Y. pestis wasn't acting alone, and given the right conditions, the modern version could once again become dangerously contagious.
So Hannah, what are you working on?
I'm working on burial cut. We think it's oriented east-west because of the discoloration. And as I was cleaning above it, this little cross flew out.
You found a small cross.
Yeah. We asked Antonio, and he thinks it's medieval and possibly used on the clothing, so.
As a pin of sorts.
Yeah.
Yeah, that's fantastic.
Medieval is music to Hendrik Poinar's ears. That could mean 1347. The year the Black Death arrived in Italy. He has come to the dig here at the Church of San Pietro, hoping to find more samples of Y. pestis.
Since we're still on this quest, this eternal quest to understand the evolution of pathogens, and in particular Yersinia pestis, the hope is that we'll find either victims of the Black Death or even later epidemics.
Although he's found nothing conclusive so far, Poinar has picked up another clue. All of the Italian chroniclers noted how rapidly the disease spread from person to person. Poinar believes Y. pestis killed so fast because it wasn't acting alone.
It was a bad winter, excessive rain, so there's a poor crop yield. And this is at the same time that you're just coming off a population expansion. So you have a large population. All of a sudden you lose your majority of your food sources from wheat and rye. And all of a sudden you start this period of starvation, right? And then as you starve, you weaken your immunity. It was a perfect storm of multiple events taking place, which ultimately led to this mass pandemic.
To find out if other deadly infections were involved, Poinar sends DNA samples from Black Death victims in London to the Lawrence Livermore National Laboratory outside San Francisco. Technicians here are on the lookout for anything that might be used in a terrorist attack. Anthrax or ricin in an envelope, sarin gas in the air, and yes, even the possibility of plague as a weapon of mass destruction.
It's fairly common knowledge that the US, Japan, and the former Soviet Union weaponized plague in World War II. There were biodefense programs that were looking at the virulence of plague and for weaponization or defense programs. but I can't comment too much further than that.
Not surprisingly, there's a certain reluctance to give away too much. Despite that, it's one of Lawrence Livermore's newest anti-terrorism devices that's helping Hendrik Poinar test his theory that Y. pestis wasn't acting alone. On this glass slide, inside a space no bigger than a thumbnail, there are more than 5,000 DNA fragments. Using this extraordinary device designed by the lab, biochemist Crystal Zhang can identify any one of 5,000 dangerous pathogens in less than 24 hours. What she discovers in the samples from Poinar's Black Death victims is fascinating.
We identify Yersinia pestis, but we also identify a Clostridium tetani, which is a bacteria that's the cause of tetanus.
That means it's possible that tetanus was a co-infection in Black Death victims.
I mean, there's a list of other bacterial co-infections. So not surprisingly, she sees things like Mycobacterium involved in tuberculosis, right? So yeah, it's sort of like we thought, in the sense that these individuals weren't sitting there with nothing else going on.
TB and tetanus still exist today. Did these deadly pathogens help turn Y. pestis into the super killer described by the Italian chroniclers?
Once we have a better understanding how a pathogen can evolve, how the virulence mechanism can change, that will help us better counter future threats.
For Poinar, one of those future threats is a re-emergence of the Black Death. If this more virulent version of the disease came back, could we cope? To find answers, Poinar consulted with Canadian biochemist Jerry Wright.
What we have done is develop a computer program that analyzes genomes to look for genes that confer resistance to antibiotics. And we're actually quite pleased that it turns out that the genome of the Black Death contains very few antibiotic resistance genes. That means today that if such a pathogen ever emerged, we would be able to treat it with a very old antibiotic called tetracyclines. So you could use tetracyclines now to basically stop the Black Death in its tracks.
But there's still a problem. Even if we can stop the Black Death now, it is continually mutating.
So there's a constant change. You get all these novel mutations. For example, flu in pigs and birds, and all of a sudden you get a recombination, and it jumps to humans, and then it's horribly deadly.
To protect us from an evolution of the disease that could kill again on a massive scale, Poinar also needs to understand how ancient Y. pestis spread. To do that, he turns to another colleague at McMaster University in Canada. David Earn is a mathematician. His specialty is creating animated models that reveal how infectious diseases spread.
It's a very important process in allowing us to determine whether we're on the right track in understanding the mechanisms that gave rise to the disease spread, as opposed to just having a look at the epidemic curve and seeing how many deaths there were each week and so on. If we want to predict what a new epidemic of a disease would be like, we have to understand the mechanism.
However, there is very little hardcore data for the Black Death in London. Unlike their Italian cousins, the 14th century English were not particularly good at keeping detailed records. But there was a second massive outbreak, the Great Plague of London. It engulfed the city 300 years later in 1665. By this time, all deaths were recorded in documents called bills of mortality.
The bills of mortality were weekly records that list the number of deaths by cause in the city of London as a whole. And during plague epidemics, they also listed for each parish the number of deaths attributed to plague. And that's why we have a detailed spatial record of the Great Plague of London.
The bills of mortality have been preserved at the Guildhall Library in London. a stone's throw from the Church of St. Giles, Cripplegate. The causes of death cover a wide range, from the incredibly detailed burnt in his bed by a candle at St. Giles, Cripplegate, to the utterly baffling.
There are things that we don't know definitely what they meant. For example, they list people dying of teeth. Well, what does that mean? Well, either maybe a child died when their teeth, at the time when their teeth was coming in, and they attributed it to the teeth causing it, or other things that, was it dental abscesses that were causing people to die from teeth.
But the bills of mortality changed dramatically as the plague swept through London. The Church of St. Giles, Cripplegate was just outside the city wall, in a parish that was densely populated and extremely poor. The mortality rate for the City of London was about 1 in 5, but here in St. Giles Parish, it was 1 in 2.
I think it's almost impossible to read through the bills of mortality, even though, in essence, they're statistics, and not feel for those people, not connect in some way to what they were suffering, and it takes you back to their time because it's describing illnesses that we're not always familiar with now, thank goodness.
Believing that the plague was spread by animals, the City of London issued a sweeping order.
That dogs, cats, conies, that's rabbits, and tame pigeons be destroyed about the town, or to be kept so sparingly that no offence may come by them, and that no swine be permitted to range up and down the streets as they frequently do, or rather, not to keep any at all.
For David Erne, it's the numbers that tell the story. By analyzing the raw data contained in the bills of mortality, David was able to create a model showing exactly how the plague spread. Parishes on the outskirts were affected first. The walls surrounding the city did keep the disease out for a while. But during the week of June 13, 1665, someone infected with the plague entered the city through Cripplegate, penetrating this last offense. Once inside, the disease spread with lightning speed. Being able to see that spread gave Poinar a totally new perspective on the plague.
And so our interest was very much in looking at differences that we see in these rate of spread of potentially Yersinia pestis causing agents across time.
But the 1665 plague of London told Poinar something else. It revealed the potential of bubonic plague to mutate and re-emerge. It did it then. Could it do it again?
This is this million-dollar question about, for example, the 1665 plague of London. Are we talking about re-emergence of a slightly evolved version of this ancestral Black Death strain? Do we have convergence in separate lineages every time this bug emerges in humans? Or is it just that Yersinia pestis has this massive outbreak, then dies down and remains in a residual population of rodents or in the sediments, sort of in a dormant state for long periods of time? But maybe that reemergence is just basically waking up and coming back out, right? So these are million-dollar questions.
And Hendrik Poinar isn't the only one asking them.
Whether plague is re-emerging or emerging, it depends on who you talk to and where you are. And because plague bacteria can remain hidden in the environment, just circulating among rodents and their fleas for long periods of time, you may see no obvious evidence that plague's there, but it could still be there circulating at low levels among the animals and their fleas, and then occasionally spill over to and cause an outbreak. In that case, some people would say it's re-emerging.
At this stage, Hendrik Poinar has a pretty clear understanding of the mechanics of the Black Death, how it spread, and how it quickly decimated its victims. And yet, there are still critical questions that elude him. The Black Death killed almost half the population of Europe. Why didn't it kill everyone? How did the other half survive?
Now, this is Christchurch Spitalfields, and they found lots of very interesting individuals from the 18th, 19th century. This is Spitalfields Market, and over there's the development, and that's where the cemetery was and all the individuals. All the many thousands.
Hendrik Poinar has given bone expert Sharon DeWitt a new mission. To complete it, she's come to Spitalfields in London's East End with Jelena Bekvalak, curator at the Museum of London.
So if you just like to look down there, you can see the remains of the medieval building.
Wow, that's really neat.
Yeah, not something you're already sort of expecting to see somewhere like this.
Sharon's job is to find bones from before and after the Black Death.
Right, so if you just come through here...
It's the first step in determining why some people survived the deadly epidemic and some didn't. When developers excavated this area during the 1990s, they uncovered a massive cemetery and an ancient charnel house. In places where land was scarce, like the center of London, cemeteries were often reused. Skeletons were exhumed and more efficiently stored as a collection of bones.
So, Sharon, this is quite remarkable because this is the medieval building, the chapel and the charnel house that was excavated. So it's quite remarkable how much is still left extant.
It's really well preserved. So how many people were excavated?
14,000 individuals were excavated. Yeah, really large number, and we curate 10,500 of those individuals at the Museum of London.
Okay.
What's really interesting is they're very well phased for dating.
Okay.
So you're able to say you've got individuals from 12th, 13th,
Oh, okay.
14th, 15th
So you'd
century.
be able to identify people who died before the Black Death and those who died afterwards?
Yes, you can.
Okay, so that's perfect for me then.
Yeah, absolutely remarkable.
The Black Death was one of the greatest natural selection events in history. It was like a macabre lottery. If your number was up, you died. But for those who didn't, was it a matter of survival of the fittest, or were they just lucky?
Well, if you kill off 50% of the population, maybe the humans were just highly susceptible. And so we actually weeded out 50% of the population that had genes within us that made us highly susceptible to plague. And the other 50% that survived had genes that made us actually protective against the plague.
Right, Sharon, this is one of the individuals from Spitalfields, so thought we could have a look and see if there's anything that might be useful.
The skull looks really nice.
Yeah, that looks like a fantastic skull.
Once again, Sharon is searching for perfectly preserved teeth.
And the point is to determine if there are certain characteristics that allowed people to survive the epidemic that they then passed on to their offspring and so forth.
Sharon sends her Black Death samples back to McMaster University in Canada for analysis. What they'll be looking for are genetic variations. Known as alleles, these may help decipher the disease. If Poinar and his team can crack this final piece of the mystery, determine exactly what shielded 50% of Europe's population, this knowledge could have profound implications for dealing with other infectious diseases in the future.
And the question will be whether or not those alleles are still of use for other things. Because maybe that protection doesn't just work for plague, maybe that protection works for things like HIV or smallpox or so on and so forth.
Back at the dig site at the church of San Pietro near Florence, Poinar's patience has been rewarded. The team has excavated remains that appear to be Black Death victims.
Yeah, no, absolutely. This is exactly what we came for, so we're exceptionally excited. I mean, it's fantastic samples.
Everything is carefully logged and shipped back to the ancient DNA lab at McMaster University in Canada.
So the good news is we have a medieval burial pit. We've sent samples off, and we're going to wait the results. And based on the screening and this enrichment procedure that we have, we should be able to determine whether or not these are victims of the Black Death. Yeah, it's a never-ending story.
People have said at various times that infectious diseases are probably behind us, and they're always wrong. There will always be more epidemic outbreaks of various pathogens, because even if we were to eradicate a pathogen completely, as we have with smallpox, for example, there are always new things that are evolving and that have the ability to invade human populations and cause epidemics.
Hendrik Poinar and his team have solved one of the greatest mysteries of science. They've identified the most lethal killer in history, and they've reconstructed the entire genome of an ancient bacteria, something that was once considered impossible.
The most interesting thing about the genome of an organism, an individual, is that the entire sort of recorded history of that organism and its ancestors is stitched into the genome of these individuals in present day. So it really represents what I see as sort of one of the greatest archaeological projects of all time.
Unlike TV crime dramas, where DNA can crack the case before the end credits roll, real life and real science are somewhat different. Poinar and his team have solved a great part of the Black Death mystery, but there are still many unanswered questions. Perhaps one day they'll have all the pieces of the puzzle. Until then, they'll keep on digging.